So RNA processing, we got this whole RNA ribbon here, but like I mentioned before, it's got parts of it that we don't actually need. We used to call the sections of DNA that were going to be cut out non-coding. We used to call it junk DNA. We don't call it that anymore. Now we call it non-coding DNA. Because it turns out it does have some functions in terms of regulation of gene expression. It just doesn't code specifically for polypeptides. It's not amino acid information. But when we transcribe a gene, we get the whole gene, which means that it's got a whole series of sections, which is coding. So this part codes for polypeptides. And this next one is non-coding. And then coding. Again, the coding sections are called exons because they will be expressed as proteins and they get to exit the nucleus. So exons. What an excellent question. Introns are the segments of the mRNA molecule that we just made in transcription. They're the segments of the mRNA molecule that do not code for anything. So let me adjustments. We're just going to make this one slightly different color there. So this section we need to snip out, and that's what RNA processing does. We go up to this RNA molecule and we're going to say, oh, okay, here's a coding section. We're going to take that, and then here's another coding section. We're going to take that, and we are going to splice them together. That means combine them together. The word splice is from if you take two sections of rope, you're like a sailor or something, you need to combine two ropes, you open up the ends, you unfurl them, and then you weave them together to create a single rope. So splicing an RNA molecule, you're just combining these two sections, right? So we combine the exons, and then we cut out the introns. The intron is removed. They are snipped out. They're snipped out by a complex of RNA and protein, which is called the spliceosome. And now you guys know that whenever you see some, it just means body or like object, essentially. So it's the object inside the cell that splices together the exons, the spliceosome. Okay? So we cut out the introns and we splice together the exons. I always remember that the introns stay in the nucleus. In the nucleus. This is a Photoshop file. I think it's missing from your guys' slides, but it's in the videos on YouTube. I can add the slides up, but it should be in the gene expression stuff. I'm not sure why it's not. In the nucleus. And then to that mRNA that we just spliced together, we are going to add a little section, a new section of RNA, which is called the 5' cap. and then another section, which is called the 3' poly A tail. And it's called poly A again because it's just adenine, 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 all the way down the line, very, very long. And then now this whole thing, this completed mRNA molecule, that can go to the ribosome for translation.